morning, good morning, good morning, good morning. I listen to a lot of dark ambient music. It keeps me moving, whether I'm working outside or just trying to think or sleep. For some reason, it's just a soundtrack that just goes on in my head. So people's shows should reflect them, even if it's not necessarily what the audience always wants. Doing what the audience wants, of course, will get you the clicks and so on, the popularity. I mean, what you see is what you get. And if you ever meet me in person, you shouldn't go, wow, he's so different in person. Welcome to the show. It's 9:39 a.m. morning time from, Massachusetts, Southborough, Massachusetts, just between, you could say it's between Boston and New York City. My name is Mark Kulak. It is October 19th, 2023. But I kind of just wanted to take the opportunity to spend like the next hour. And this is, it's really an update. So the title of this video, and it's good to see Kim here. It's good to see Christy here, is Acuitas and AbCellera, or AbCellera, and AB stands for Antibody AbCellera. These are two biotech companies in Canada, based in the Vancouver area. And the reason why we're covering this is a recent broadcast from Jonathan Cooey that I mentioned several times, where he highlighted a video from a Pieter, spelled Pieter, Cullis, from the year 2022 University of Manitoba Gardner lecture by Dr. Pieter Cullis, who is right there. And this 1 hour lecture, it's a 1 hour lecture with a couple of questions and answers. Dr. Cullis goes over the company that he has been working on, his life's work, and so on. And the company that he has been working for the last several years is a company called Acuitas, Acuitas. Sometimes I misspell it with a Q. I'm, for some reason, compelled to put a Q in there, but I'm pronouncing it Acuitas. Maybe it's Acuitas, but Acuitas is how I've been saying it. And the problem that they have been solving is these are chemists, and the penetration of cell membranes, providing technology for that. And as you can imagine, this is a key technology which has been utilized in our modern era for the products which have been manufactured by companies like Modna and Pfizer for delivery of messenger RNA into cells to encourage cells to express proteins. Proteins which are then which are described as antigens, or an antigen similar to the coronavirus, right? And in response to that, as has been described by us, the antigen which is produced, the foreign protein which is produced, will encourage the body to make antibodies to that, using the body's natural system for making antibodies, which again, antibodies themselves are just proteins. You follow me so far? Now, there's several shocking revelations made in this video, which we, thank goodness, Jonathan shared this with us and played it live. But before I share those, I will remind you that I've seen this name before, Pieter Cullis. I haven't really researched him that extensively. But the reason, in case you're wondering, the reason why we're looking at Pieter Cullis, the reason why we're looking at him is because a Dr. Robert Malone, who requires no introduction on my channel. There is only one Robert Malone who's ever lived of serious significance, or at least with respect to the content we've covered, is Malone, in a recent interview, said that he did not believe that, I think it was recent, it might have been a year ago, that Drew Weissman and Carrico, the two people who were awarded the Nobel Prize for their work on the lipid nanoparticle, or the technology which made mRNA vaccines available. If anyone should have won it, other than, of course, Robert Malone himself, it should have been Pieter Cullis. All right, that's cool. So that was Malone's point. So having said that, That doesn't mean that Cullis is a good guy or bad guy, legitimate or whatever, but for some reason, I'm curious as to why we're told to look over there. Cullis in this video gives lots and lots of shout outs to both Weissman and Carrico for their work on the lipid nanoparticle. It sounds as if he might have wanted to also be on the stage with them. Like maybe he should have felt as though he should have also won a Nobel Prize. Okay, fair enough. And then he gives shout outs later on. One of the most shocking things that he says in this video was that there was no way to control where the mRNA went. He said it, and he is a key person with respect to the technology of this delivery vehicle of messenger RNA. No way to control it. He said, I burnt 5 postdocs. In other words, postdoctoral students working for him. He burnt 5 postdocs, tasking them with trying to find a way to control where the injection goes or stays. Couldn't do it. And a 6th one threatened to leave if they didn't get a new task as a postdoc student. And I guess he allowed them that 6th person to take on a new task. So he was unable to do it. And this is 2022, this presentation. So while everyone else is being told that it is conspiracy theory to think that the mRNA shots don't stay in the arm, for everyone else, Mr. Serendipity was mainly concerned with controlling where the money went. Yeah, we'll add to that. Good to have you here. And while it was conspiracy theory to suggest it wasn't, this guy said on the stage that there was no way to control it. The bone he threw was that maybe someday there'll be a technology by which we can select which tissues, which organs uptake and don't uptake the mRNA. Now, that's amazing. That is quite the confession. That means this stuff's going all over the place. Okay? That doesn't mean that's not good or bad, but it's definitely counter to what people have been told and told to believe. And he's one of the experts. Now, another thing that he talked about in this lecture was something called silencing RNA or interfering RNA. And you know what messenger RNA is, right? It comes into a cell, and this is how it's been described to us. The cell welcomes it in, and from via essentially a extracellular vesicle, exosomes or synthetic exosomes, and then the cell, in response to their constructs a protein, right? So the messenger RNA is a description of a protein to make, and somehow the cell knows how many to make in response to it. I mean, you just make one. Do you make 1,000 per mRNA? I don't know. Either way, that is how this messenger RNA technology has been described to us. But there's also something called silencing RNA, and this is where RNA delivered to a cell somehow controls which genes are upregulated or downregulated. In other words, you can supposedly silence genes. You can turn genes on or off. Presumably, this is methylation via silencing RNA. All right. I've never really, I didn't know that technology was out there, but that technology, the silencing RNA technology, has been in the works for 30 years. There were companies in the 1990s that were looking at this stuff. Gene silencing. Okay? Hey, good to see you, Rhonda. Good to see you, end of days. All right. So, and in this discussion here, Peter Cullis extensively, but for a good section, talks about his background with silencing RNA, silencing and interfering RNA, siRNA. And the timeline he gives for that is 1990s and early 2000s. I found that to be incredibly interesting because, and as we said, it was Robert Malone who told us to look at Peter Cullis. All right? And if you look at Robert Malone's resume, in the year 2000, he had a senior role with a company called Intradyme, and which combined with another company called Silence Therapeutics. Let's take a look here. And then we're going to go to the Malones. So Malone was a co-founder, one of 3 co-founders for a company called Intradyme. He was the CSO, Corporate Science Officer, a board of director member from 2000 to 2001, Intradyme Corporation. According to Malone's resume, Intradyme was a biotechnology company that develops gene therapeutic technologies based on RNA interference. Intradyme merged with Silence Technologies in 2009, and the merged company is now publicly traded. And this is as of 2017. Silence Technologies is involved in development research of targeted RNAi therapeutics, RNA interfering therapeutics for treatment of serious diseases. Dr. Malone co-founded and helped secure $2.3 million in venture capital funding, including monies from the Novartis Venture Fund, ETP Venture Capital Fund, and the state of Maryland. University of Maryland, just think Gallo, etc. Performed facility setup, infrastructure setup, and intellectual property development. Intellectual property development. Another way of saying patents. Business and technology development planning, including in-depth business and scientific plan. Now, first off, and I won't speculate too much more on this, at the same time, he is a co-founder and a board director, a board member of Intradyme. He's also with the United States Uniformed Health Services, setting up a breast cancer tissue bank laboratory in Windber, Pennsylvania. This is the Joyce Murtha Breast Cancer Research Facility, which is part of the Windber Medical Lab, where they were going to effectively have a high-speed data link to Walter Reed Medical Center for studying, analyzing data on cancer in the U.S. military. Interesting that Malone would be involved in such a project and getting access to cancer in the U.S. military just before the time that there was potentially a planned anthrax attack, which was going to require a new generation anthrax vaccine to be used on the U.S. military. This would, if one were to be conspiratorial, this would look like, wow, Malone knew that there was a chance of cancer with this new therapeutic, which was going to be rolled out because the existing anthrax vaccine was just so bad. They just made people take shot after shot after shot after shot after shot. Of course they got sick. So this overlap is quite interesting. I've not dug into it too much. Intradyme is a very difficult company to get data on. I have some articles. It's an incomplete picture. And I started to look into them like a year or two ago, and I only went so far because it just hit a dead end, and I wasn't really sure what it meant. What is this silence therapeutic stuff? Okay, so going back here. So he mentions, Cullis mentions RNA silencing, gene silencing. So I'm not really sure where the next step is for this Acuitas company, all right? What's on their roadmap next? How are they going to get their next big money payout? What I do know is that they have this background in gene silencing and silencing RNA. Now, if I may, I had 2 tweets earlier. This was a research paper from 2012. Maximizing the potency of silencing RNA lipid nanoparticles for hepatic gene silencing in vivo. And again, this shows that we have the Peter Cullis, Thomas Madden, and so on. These people who are at this Canadian biotech company, Acuitas, looking at this technology. And what I found, and Peter Cullis okay, Peter Cullis, this guy here, there's two people who really co-founded this Acuitas company, Acuitas. One is him. The other is a very difficult, actually even harder to get information on, Acuitas, and that is Thomas Madden, Madden. Now, Peter Cullis, his family roots are in a couple of generations ago in England, but the family is a little bit less connected with the UK. But Thomas Madden was born in the UK. He is British, and he came to Canada in the 1980s, I think. He was born, I calculate, in 1954, but I have not been able to get his birth date just yet. Okay. Malone says, if anyone should win the Nobel Prize, it should be Peter Cullis. We look at this company, Acuitas. Peter Cullis's longtime peer at Acuitas is Thomas Madden. Cullis and Madden have been working together for 40 years. They started working together in 1982. They have a 40-year collaboration, which starts with companies like Techmira, and it just kind of evolved from there into Acuitas, right? But for 40 years, they have been working on this technology for getting RNA, getting genetic material into cells. And of course, sometime around the 1990s, that connects with the fame and the money of gene therapy, because gene therapy was really picking up in the 1990s. I think Peter Cullis was at one point a very legitimate bench scientist. Built all his own equipment. You could tell he knew the math. He knew what he was talking about. And at some point, I think the money and fame kind of got to him. So the other guy there is Thomas Madden. Okay. Now, Thomas Madden, believe it or not, just 2 days ago, October 17th, 2023, this Thomas Madden was on an interview with Chris Spivey and I guess this is farmtech.com. 2 days ago with Kate Broderick. Kate Broderick, who is the bigwig previously at Inovio. She was a senior vice president of Inovio. Now, she's no longer at Inovio right now. She is with another company. But let's think about this here, okay? Let's follow this through. Robert Malone says that of all the people who should win the Nobel Prize, other than him, of course. Matter of fact, he probably believes he should win every Nobel Prize from this point forward until the day he dies. He might actually believe that too. I don't know. But would be Peter Cullis, the guy that Jonathan Cooey introduced us to with this video. And I think this guy's a nut job, and a bit of a monster for still pushing Zika stuff. But Peter Cullis's 40-year Acuitas, 40-year peer is Thomas Madden. Thomas Madden 2 days ago has an extended interview where the other guest is Kate Broderick, who is the former vice president of Inovio. The very same freaking company that Malone says he co-founded himself. And putting the pieces together, what it looks like is that this lipid nanoparticle system to deliver RNA silencing, the lipid nanoparticle technology for RNA silencing, sometime between 2011 and 2013, was repurposed for the delivery of messenger RNA. This gene silencing technology, this silencing RNA technology, which has been worked on since the 1990s. Here, this article is from July 10th, 2012. 20-something years later. Robert Malone was a co-founder of Intradyme, which became Silence Therapeutics, which did a lot of work with Cullis and Madden. No doubt. This technology around 2010, 2012 was repurposed for the delivery of messenger RNA. Now, here's the problem. Why was it repurposed? 2010 to 2012, all right? I wish I had a timeline available. I got so much data right now, and I got so many things to do. And as you know, I'm Mr. Mom right now because we're raising our grandchild now full time. So the people who don't want this information out there should be counting their lucky stars that this is the situation for their own sake. Otherwise, we would have a whole lot more stuff written up by now. But why was it repurposed? Well, first off, there was no really super valuable therapeutic value of gene silencing. Maybe there could be someday. You could speculate there may be, but they couldn't find one and they couldn't get it to work well enough. So why in 2010, 2011, 2012, the urgent rush to repurpose this stuff, to redeploy it for messenger RNA? And messenger RNA, instead of just this RNA being used to I guess, affect methylation and to turn genes on and off, right? Instead, this RNA is being received by cells, and the ribosomes are processing it and creating proteins which are considered antigens. It's reusing a lot of the same technology, but now instead of the point being gene silencing, we're having the cells make proteins directly, which are described as antigens. Hey, McDuff, I already told my audience I'm definitely finishing by 11. I don't believe in streaming over people who I really admire like you. That's a big question. Who silenced my P53? Well, here's a great question. The technology, the gene silencing technology, okay, the gene silencing technology here, the silencing RNA, which has been looked at by companies like Acuitas, which go back to this Peter Cullis and Thomas Madden, was also part of Robert Malone's background, because he was a co-founder, one of 3 co-founders, chief science officer, board of director members of a company called Intradyne, which also did RNA silencing. And eventually their Intradyne merged with Silence Technologies in 2009. And as I just said, if you don't think this long-term association with Malone isn't real, here we have Malone says he's a co-founder of Inovio. And here we have the previous senior vice president of Inovio, Kate Broderick, 2 days ago on an interview with Thomas Madden, the co-founder of Acuitas. There is definitely a loop going on here. There is definitely a very serious loop going on here. I know I'm putting a lot out there, so you're going to have to watch this one a couple of times. Hopefully I'm trying to find a good cadence of repeating things. So why the pivot in 2010, 2012 to start repurposing 20 years of technology of delivering RNA for gene silencing into using RNA for messenger RNA, or using that technology for messenger RNA? Well, 2010, 2012, this new technology, the way they're going to be using it now, is they're going to use it to encourage cells to make a protein. Now, the way the Pfizer vaccine works, the way the Moderna vaccine works, is that it's this, we're told that these products make an antigen, okay, a foreign protein, an antigen that has some association with the SARS-CoV-2 virus. It's the spike. And then your body makes antibodies in response to that. Okay, we've seen this diagram many times before. What Peter Cullis also says in this interview, in this presentation in 2022, it says, you know what, guys? At some point, we don't need to make antigens with this messenger RNA. We can just start making the antibodies directly. Why inject the mRNA? Cells uptake it. Some cells do. The cells make the antigen, and then the body makes the antibody to the antigen. Well, antibodies are just proteins, right? Why have the middleman? Just cut out the middleman and make the antibody directly. So Cullis is laying out the roadmap of where messenger RNA technology is going, is to just make the antibodies directly. Get rid of the middleman. Make the antibodies directly. You follow me? It was sometime between 2010 and 2012. One of the most important protein patent disputes of the last 20, 30 years happened, involving a company called Centocor, and the other one was, I believe, Genentech. This was, I think, one of the final straws to be pulled, where it became very clear that the ability to patent antibodies was going to get impossibly hard. This is the antibody patent paradox. As we've learned more about proteins, as our ability to describe proteins has increased, we've realized to describe them in greater specificity, the more we can describe, the more we realize we don't know. They have found no consistent correlation between antibody structure or molecular description and function. They can do this, exactly the same antibodies can do different things in different humans. Different antibodies can do the same thing in different humans to some extent. It's not absolute, of course, but to some extent. Hence, hence patenting this stuff based upon function and form is becoming increasingly impossible. Now, there's antibody patents from 20-something years ago, 30 years ago, based upon what was understood to be the state-of-the-art at that time. But what was understood to be at that time as there's ranges of antibodies doing certain things in all people has turned out to be science fiction. It wasn't meant to be science fiction, but it turned out to be that way. So if you can't patent antibodies, and the biggest sector and growing sector of pharma has been the manufacture of antibodies, $160 billion a year. How do you get around this? How do you get around the issue? I wish you could see my notes. How do you get around the issue? Let's go to the chat here. This is the biggest takeaway here. We're going to get into Palantir and Peter Teal. Really, this stuff gets wild. But how do you get around the inability to patent antibodies? How do you get around that? If you want to keep the research going, if you want to keep the money pipeline going, how do you get around the fact that these antibody patents are getting impossibly hard? How? The answer, I don't see the answer in the chat. The answer is you forfeit manufacturing antibodies directly. The answer to the antibody patent paradox is to have the body make the antibodies directly. This way, you don't freaking need to patent them. The way to get around the antibody patent impossibilities is to just use mRNA, to just use a technology that causes people to make their antibodies themselves. And this way, you don't need to deal with all the intricacies of structure and form and molecular description. You get around it. You've gotten around it. And what Peter Cullis here is saying, what Peter Cullis here is saying, guys, is the next generation of mRNA, there's going to be no antigen. You're going to go straight. You're going to go straight to the antibody. You see that? And as we're showing here, we speculate that at some time between 2010 to 2012, the same time that the patent issues with this very intriguing company, Centocor, were wrapping up. And by the way, the lawyer involved in there giving legal advice, I believe the Centocor, was Les freaking Weinstein, father of Brett and Eric Weinstein. That patent case with Centocor was inspiration to say, you know what, we need to find a way to deal with this patent issue in antibodies. So they pivoted 20 years of research and silencing RNA into messenger RNA to cause the bodies to make people's bodies to make their own antibodies. The timing all lines up perfectly. It's perfect alignment. Meaning the Weinsteins must have known about this. Their father, Les Weinstein, obviously knew about this with Centocor, with being a co-founder of Intradyne, which became Silence Technologies, and a long-term collaborator to some extent of Cullis and Thomas Madden. We know that Malone definitely knew about this, definitely knew all about this. We know that Moderna knew about this. I mean, heck, when was Moderna made? Wikipedia, Moderna. Moderna was made in 2010. Now, Moderna, I think initially didn't think that they would be focusing exclusively on messenger RNA for the purposes of protein expression. I think that they were there was going to be perhaps more use for the signaling. But they came out of nowhere in 2010 with a ton of money, with Rossi and Springer. Timothy Springer, by the way, also has an indirect connection to Robert Malone via Washington University in St. Louis. Langer, now Baratheon, MIT. All these companies kind of popped up in 2010, 2011, 2012 with the pivot of this gene silencing technology, which still needed more work. However, they were most of the way there at having a product, an RNA product, which could be delivered to encourage cells to do something. It was the closest thing possible, the closest method possible for getting the human body to make their own antibodies and getting around this absolute catastrophe of antibody patents. This is why they wanted to use this technology. This is why they pivoted so hard and so fully to this technology. Those patent issues, it's not just the money. Remember, whoever's controlling antibody patents has one of the most powerful hands in the world. This biomedical technology is one of the most powerful things on Earth. I would say it's as powerful as the entire fleet of United States aircraft carriers. This is extremely rich, extremely powerful technology. It doesn't always work. As a matter of fact, I would say most of the time it doesn't work. However, the promise of it working is still there, possibly, in some cases. The ability of it to do harm is definitely there. So this isn't just a couple of guys wanting to make some extra money protecting their patents. This is a massive geopolitical investment, a strategic investment, as much as oil, diamonds, hydro. This is serious tech, which is one of the reasons why I've been calling out the number of people who seem to have communist affiliations involved in taking it over within the United States. So, what year circa was the antibody patent? 2010, 2011, 2012, wild tangent. In fact, you can look at, let's see here, Centocor protein patent. Just type in 2011, and you're going to get tons and tons and tons of articles. GmbH versus Centocor Orthobiotech, patent infringement involving a class of antibodies developed to beat certain autoimmune diseases. the amount of money involved was not stellar. Okay? It's not, do not get caught up in the dollar amounts in this topic. This is of massive geopolitical strategic value. And Centocor, by the way, as I've shown in another video here, do I still have that slide share up? I have a little bit of a person map here. Very, very important person map. Centocor was co-founded by Hillary Kaprowski. The lawyer, I believe, representing Centocor, I know he was involved in the case. It's on his own CV, so I know it's not a lie, was Les Weinstein, father of Eric and Brett Weinstein. Kaprowski was a co-founder of Centocor. Hillary Kaprowski, and this should interest you, McDuff, because Kaprowski is one of the three people making those polio vaccines. Okay? The other one was Jonas Salk and Sabine. At the Wistar, he was a longtime director of the Wistar, and two of his key students were Dave Weiner, or proteges were Weiner and Plotkin, Stanley Plotkin. Plotkin is the rubella guy. Dave Weiner wound up becoming the next director of the Wistar Institute in Philadelphia. And Dave Weiner and Stanley Plotkin, Dave Weiner then became a co-founder of Viral Genomics, which was rebranded Inovio after they bought the Inovio up in Norway. Stanley Plotkin was a chief science officer in Inovio, and Robert Malone claims to be also a co-founder of Inovio. All very, very interesting because 2 days ago, we have the previous senior vice president of Inovio, Kate Broderick, who talked with the very, very strong Scottish accent on an interview with Thomas Madden, the co-founder of Acuitas Therapeutics, 40-year peer of Peter Cullis, the guy that Cooey just did a video about. Within 2 hours of Jonathan Cooey doing a video about Peter Cullis, I had two people who I deeply respect, I won't say names, contact me and say that they thought Jonathan Cooey was a fraud. Out of nowhere. Something about Cooey talking about Peter Cullis alarmed some people who I still deeply respect to think that Jonathan Cooey was suddenly a total fraud. Now, I totally, depths of my soul, disagree with that position. However, that reaction was enough to make me go, wow, Cooey is really on to something now, because there are some people who are really upset. I should dig in a little bit more on this. Every time I see people respond, thank you. That's the metadata that I need. And again, fortunately, I'm caught up in the life of Mr. Mom right now, so I'm missing a lot of stuff. You should be thankful. So now that we know Peter Cullis and Acuitas is a sore spot, let's dig in. We know from our mind map here that Robert Malone does his premiere, his intro to the world, on the Dark Horse podcast with Brett Weinstein. This Dark Horse podcast, the whole intellectual dark web, was started in 2017, inspired by Eric Weinstein, brother of Brett and son of Les. So therefore, we know that there's something about the Weinsteins that is really important to know. Now, we know that Eric Weinstein has made a ton of money because he became an advisor, a financial advisor to whom? Peter Teal. And I did a few videos introducing this concept of Eric Weinstein, who I'm sure has some amazing inside information, given that his father is Les Weinstein in the monoclonal antibody industry. We know that Eric Weinstein cleared at least $100 million and has been working with Peter Teal. All right. In 2020, November 2020, Peter Teal decides to make a massive financial investment, buys 5% of the company stock-wise, and gets on the board of another Canadian biotech company called AbCellera or AbCellera. AbCellera Biologics in Vancouver is a Vancouver, British Columbia-based technology firm that researches and develops human antibodies. The company is best known for its leading role in the pandemic prevention platform, a project of DARPA's Biological Technologies Office. Okay. Now, they, again, founded 2012. Now, their main founder is the main guy that keeps coming up is this guy. His name is Hansen. I think, and he's the total stage corporate type. You know, I think he shaves his hair, perfect teeth, perfect skin, perfect coat. I mean, it's just like his goal is to look good on a stage and present a product. This company, AbCellera, what they do, they're effectively a big data company where they do is they search for antibodies. So in other words, if there's a new outbreak of something, according to the narrative, you go through the blood samples and you find the antibodies that are in the people that have survived the illness. So you use big data to find the antibodies. You don't design them, you find them. You don't have to patent something you find. You only have to patent something that you design, right? And AbCellera, or AbCellera, was the first company to make an antibody for COVID. They were the first. The product was called Bamlanivimab. I keep wanting to say Bamlanivimab. Bam, balam, bam, blam, bam, 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 bam. Make antibodies wild, bum, ba, bum. You know the song, anyways. Or LYCOV555. And this was an antibody treatment that was made in record time, way before Faster and Regeneron. A whole bunch of people received it. It received accolades of working. I don't know how they did it. I suspect total corruption, and they probably had it ahead of the whole thing ahead of time. I learned about AbCellera because they are very, very close. They're not related business-wise. They don't own each other, but they're very, very close to Acuitas, where Peter Cullis is. As a matter of fact, AbCellera and Acuitas are often equated as the two top most promising biotech company startups in all of Canada. They're both in Vancouver. And AbCellera is developing these, they develop an antibody, but the way they do it is very different now than, let's just say, Regeneron did. Now, there's this video here that we found called, Did Peter Teal Find the Palantir of Drug Discovery Software? This is a video from a year ago, and it describes AbCellera, which is not related to Celera in the United States. This is not related to the Human Genome Project. This is a Canadian company. I'm sure there's connections, but did Peter Teal find the Palantir of drug discovery software? And so Peter Teal becomes a board member, 5% owner of AbCellera, which does what? It mines people's blood samples to find antibodies. Antibodies which are then somehow manufactured and given to people. And AbCellera had a product that worked for COVID, and then they were taken off the market and put back on the market, and so on and so forth. Peter Teal is receiving financial advice from Eric Weinstein, brother of Brett Weinstein, who introduces Robert Malone to the world, and son of Les Weinstein, who's the attorney handling this Centocor case. Now, the next thing, as we have read, AbCellera, Antibody Celera of Canada, has announced a business relationship with Moderna. AbCellera has announced that they have developed a partnership with Moderna. In September 2021, the company announced a multi-year agreement with the Moderna to develop an mRNA antibody treatments against multiple diseases. So let's play this out. AbCellera has announced that with their partnership with Moderna, they are going to have a product that can find an antibody, not design it. It's going to find it using big data, the Palantir of drug discovery. And then you don't have to worry about any patents for making the antibody because you're going to make the antibody using mRNA technology from Moderna. This is a coup. This is totally getting around every single possible contentious issue related to the patenting of antibodies. What a beautiful solution. And we know from Peter Cullis that we got the strong reaction to say no longer trust Jonathan Cooey, where Peter Cullis says, oh, well, you're just going to, at some point, start making the technology of mRNA will just start making the antibodies directly. We have that acknowledged now. We have AbCellera, which was probably encouraged Peter Teal to invest in this company by people with inside knowledge about antibody issues, such as Eric Weinstein, son of Les Weinstein, handling the Centocort case. Of course, you can't go wrong if you're Peter Teal, if you're getting this type of inside information. We know we've established that the way around the antibody patent paradox is to have people's bodies make their own freaking antibodies using mRNA. This is the org chart that you are not supposed to talk about. This is the org chart you are not supposed to be thinking about. Not Jordan Walker, not a couple of people up at Pfizer. This is the org chart you are not supposed to be looking at. And it's a very small org chart in a way because it's not really consolidated to one company. And there's no gain of function here. There's no gain of function. There's no nasty viruses. There's no mutants, none of that crap. This is all about personalized medicine. This is all about getting involved into the synthetic exosome game. This is about bypassing patents. And this is about putting intellectual property into the hands of potentially political movements that do not have the aspirations of the U.S. citizens at heart. This is the org chart. It spans several companies. This is the reason why mRNA was being pursued. This is why Brett Weinstein is so popular. This is why Robert Malone is controlling the narrative. This is why, as Jonathan Cooey so elegantly showed last night, Kevin McKernan is going to find every stupid excuse in the book to say, well, the mRNA didn't work for this reason. It didn't work for that reason. Didn't work for this reason. Didn't work for that reason. But at the end of the day, it must be shown to have worked because this is their future. They've banked everything on this. This is the Peter Teal connection. Now, the value of these stocks is not doing well. Matter of fact, it seems to be going down. Now, the reason why it's going down could be, one of those orchestrated, bad news comes out and then, you crash the market and then the people buy the stock and the value goes back up. Another possibility is that while some of these monsters knew maybe there'd be some health issues and some cancers, the other possibility is that they didn't actually expect cancer rates over the course of 3 years to explode 300%. The other possibility is that they are screwed. The other possibility is that over the next few years, people are going to be calling for blood. It's so bad that I could see there to be a desperate attempt to stage World War III just to distract from this. I know there's a lot of tensions in the Middle East, there always will be, and I don't pick sides. However, I've already seen so many millions of excess deaths around the world that were completely unnecessary, caused only by the push for this therapy via an orchestrated and organized pandemic. And there may be tens, maybe even hundreds of millions of long-term injuries and deaths as a consequence of this, which supersedes the cost paid in any world war to date because of these people wanting to control this technology. This is the biggest story. And when I see trolls in Jonathan's chat saying, Malone's irrelevant, stop talking about Malone, I'm like, there you go. Trust me, if you're talking about someone that irrelevant, eventually it's going to burn out and people will stop listening. Now, this all needs to be drawn out. Of course, we need to draw more linkage maps. I need to archive a lot more of this. I need to learn more about Thomas Madden, of course. There's still what's the Inovio angle? That DNA technology isn't going away. There's still a lot of big questions. All right. But I feel very, very, very confident that we are going in the right direction here. So let's do comments for a little bit and maybe 10, 20 minutes or so. And I don't go back I'm going to go back a little bit. Hey, Dawn New Way. Palantir with Alex Karp is the first CEO to visit Zelensky in Ukraine. Holy crap. Well, Dawn New Day is on fire here. Let's see here. Asanya, good to see you. If I recall, our former governor, Ducey, was invested in Moderna. Yeah, there's a lot of people with investments in Moderna. Of course, Moderna, as you know, mRNA is a stock ticker, right? Created in 2010. AbCellera was created in 2012. Funny how these two companies, Moderna, 2010, and AbCellera, 2010, 2012, that are now partnering on potentially the perfect solution, right? Finding an antibody, so you don't have to say you designed it, and then using mRNA, so you don't have to patent an antibody you're making. Funny how they were created at the same time, isn't it? AbCellera and Moderna. Kind of funny how they were created around the same time and eventually found each other with complementary technologies. And as a matter of fact, the part of this equation that is the most contentious with respect to the patent issue, possibly still, which is AbCellera, is over the border in Vancouver. Why is DARPA, and I'm not the biggest DARPA basher. I think DARPA is misunderstood sometimes. But let's be serious here. Why, as an American, and I know I have lots of audience members around the world, as an American, why is my DARPA investing in a Canadian company? No offense to Canada. I love Canada. But why couldn't that work be done in the United States? That seems to be kind of a big deal, this whole mining for antibodies thing. And can you imagine in the future, they can just say that they found things that were already there? Who knows what kind of crazy stuff they could be doing, right? You can design a synthetic clone, but then you can say you could say you designed something that leaked out of a lab, and then you can show people the test to say, look, you're positive for something that leaked out of our lab. But you know what you can't do? You can't get into a time machine, go back in time, and then test a person to see if they didn't test positive on it already because of something that's natural 2 years ago. You can create the illusion of spread using PCR tests and time by just testing against something that's already there or already has been there. Let's see here. Moderna spun off DARPA technology even as Palantir is spun off with DARPA tech. Yeah. Well, Moderna received, that's a good point. Moderna received, I think there was at one point $25 million grant offered by DARPA to Moderna that Moderna only used a little bit of. So Moderna was not really created by DARPA. They received some startup money from DARPA. That's a point I will defend against. However, it sounds like DARPA definitely invested heavily in this other company, AbCellera. So the point here isn't that DARPA's innocent. It's like, let's call out where they're at, where things are more and more suspicious. A little bit of investment in Moderna. They did some intellectual property sharing. I'm aware of that. But I think there was a bigger investment with AbCellera. So AbCellera mines and finds the antibodies, which is a great way of getting around saying that you designed an antibody. Of course, again, you can seed things here. You can have people, you can give people a certain infection, and they can create an antibody, perhaps something that you already knew that they're going to create. And then you could mine the blood of them and find it and say, well, I guess everyone needs this now. There's all kinds of ways that this stuff can be misused. I worked in the computing industry. I love computers. I love data mining and talking about how to use big data analytics and the pros and cons and data management and backup and archival. I think these are super powerful tools. However, as we've shown, going all the way back to the ability to predict U.S. elections on national television in the 1950s, computers can be misused to create a sense that there's an answer known that is more accurate and greater than any person could have figured out on their own. Computers can be used as one of the worst forms of deception. This is possible, and that could be just through all kinds of ways. It could be through, again, like the advertisements about the election predictions. It could be through data mining. It could be through who knows what. But computers can be used as some of the worst parts of, I'm going to say fifth-gen warfare. I don't know how else to say it. What's LENR quirky? I don't know what LENR is. I'm sorry. I apologize. Dawn New Day. Remember to investigate Factor 8 in the Clinton-Biden program to put low drug use people in corporate prisons where their blood was extracted. Oh, my goodness. Oh, my goodness. We're going to be wrapping up in a few minutes here. I don't know who you are, Dawn New Day, but you are just piling it on, my friend. I'm going to read their quote here. Remember to investigate Factor 8 in the Clinton-Biden program to put low drug use people in corporate prisons where their blood was extracted and then sold to Canada. Am I out of line in saying that while Canada is an independent nation, there's something about it that isn't totally independent? They still seem, there's some weird thing where they still have some association with the British Crown. I don't get it. I don't understand it. It's not my country. So again, I got enough problems down here, but Canada is clearly being used for issues such as this as a way to get out of some of the U.S. systems. And I see that, again, there's a lot of people with very friendly connections to, Five Eyes, British intelligence, even communism, in our picture here. I don't know how to best characterize all of it, but it's real. Hello, Allison. Jen says, have you heard of the new Institute for Health Computing, which will marry AI, patient data, and clinical expertise to improve health outcomes? No, I haven't heard of that. University of Maryland. Yep, that would make sense. That Tiger was at the Institute of Genomic Research. Is Craig Venter, and his wife is a long timer at University of Maryland. I don't know why University of Maryland wound up have so much historic significance in this story. Gallo is a long-time University of Maryland guy. I don't know if he's still active or not. And that's where Robert Malone, and his couch surfing friend David Hone both worked. 98, 99, 2000 was University of Maryland. Hey, Kim earlier says my blood is for physical, going for places and use ways you don't know of. Probably, Kim. Probably. I think all of our data has been used. And you guys, we're all free to have different opinions on this. I don't actually mind, my providing data which can be used for the greater good or by scientists. On most people don't. This can be very interesting stuff. It can be very collaborative, very beautiful. If it no skin off my back, so to say. But when it's misused, or when it can be turned against people, or when then in the future there can be new therapeutics rolled out on the population that provide no value, but wind up killing and harming people. When people are providing data, providing value, and getting nothing in return from it while others benefit, then we have a problem. Individuals who choose to make their data available, which can be used for research and some other greater good possibly, should receive some financial reward for it. Especially people should receive reward if they're exposing themselves to medical risk of via experimentation. But we all know this, right? I'm not a teetotaler here. And I think most of you aren't either. It just needs to be above board. But there's so much money and geopolitical power behind this technology that it's almost irresistible for this type of corruption to exist. The other thing here, I just noticed this with AbCellera. AbCellera was founded in 2012 by biomedical researchers Carl Hansen. My goodness, that is like the most waspiest name I've ever heard. Veronique Leco, Kevin Hayries, Daniel Da Costa, Ole Petriv. I never heard of any of these names. You can see the Carl Hansen video. You can see him. Again, he's total polished corporate type, a construct. And then these videos here. Did Peter Teal find the Palantir of drug discovery software? So I have been looking at this AbCellera company for a while. I couldn't really understand what it is they did and didn't do. It just didn't quite make sense. I think we get it now. Now we get it. Wow. And their association with Moderna. You see where it's going. But all these stocks are going down. Meaning, look, Wall Street, every once in a while, I think greed is good. There are investors on Wall Street that don't want to lose their shirt. And when the metadata says that these companies are providing products which are causing a lot of harm, there are people on Wall Street who are greedy and selfish enough that they will listen and they will respond. There's a lot of corruption in the world that can pump these companies up, but I'll tell you what, every once in a while, these greed and the selfishness of some investors can be a savior. And as the value of these some of these companies is going down, it is a reflex that something isn't quite working out. It could be one of the crash and then pump. Could be. That's totally still possible. But it's not looking good. I showed the stock to a few. I've been looking at some stocks lately of companies that do cancer treatments. And I saw their stocks go up 200% and sometime in early 2022. That means that individuals who are investing in cancer therapies and cancer diagnostics, screenings, did not actually expect that much cancer as a response to this new era of medicines. I know that there's many call that say that there's a depopulation play, harming people, possibly. But I think it's true that CDC and FDA officials, quite a few, genuinely did not expect there to be as much illness created in response to new therapies as there is. And eventually, Wall Streeters saw the data and started to invest. And it was sometime in early 2022. You can see this spike in valuations of these cancer diagnostics companies, not companies that are doing this messenger RNA crap. I mean the diagnostic ones, because at that point it's undeniable. It's getting out of control. The tidal wave is coming through. So when you see the stock of Moderna going down, let's see here. Was it mRNA, I think, is the stock. Look at that. Look at the 5-year Moderna. Oops. What? September 10th, 2021, it peaked at 450. Today, it's 82. When did I say the cancer therapy companies were spiking? Early 2022. Look at this. Almost all the stock drop in Moderna happened by February of 2022. At some point between here and here, between September of 2021 and here, 2022, it became clear a mistake was made. And instead of confessing that this medicine never should have been used and was pushed, probably for reasons related to intellectual property wars, global intellectual property wars, the influencers kept saying, "Oh, well, Fauci bad. Oh, DNA contamination. Oh, there was a little bit of dirt in the vials. Meanwhile, it was sometime around here. It was I think it was early 2020 that I think there was only one person on the internet, Jonathan Cooey, MD, or PhD, excuse me, that said we should, or maybe I forget when it was that he called it out, that we should have a pause to all these transfections. We should stop this nonsense now. And it's funny because after talking about Peter Cullis, people are now pinging me saying that Jonathan Cooey must be a fraud. Or if people only listened to him, there wouldn't be an avalanche of cancer now, would there be? If people listened to him instead of Dr. Bus Rider MD, there wouldn't be an avalanche of cancer now, would there be? Well, I've picked my alliances, guys. I don't know about you. But the AbCellera, really important. You can see where everything was going. I'm sure there's still going to be plenty of a push to go in this direction. The question is now is how bad are things and how off the rail is this whole machine? Because I think that there's a lot of unpredictability right now. And there may be a lot of attempts to distract people with other geopolitical issues and now social justice issues and rioting and so on and so forth, because things really are that bad. They really screwed up that badly. So in the meantime, stay positive. Someone gave me a good idea last night. We should have some role play discussions about Thanksgiving dinners and so on. How to behave, what to say, what not to say, how to get people to think. I don't know how I'm going to find the time, but we will definitely do something like that. I'll use some of my sales, competitive intelligence, sales playbook backgrounds, and we'll put some things together with respect to that. But with that said, everyone, thank you for joining. Great conversation, great thinking, huge progress collaboratively. I appreciate you supporters. I appreciate your friendship. And again, your patience as with everything. Get outside, get fresh air. Don't spend too much time in front of the screens. And we'll be in touch soon. I'm just gonna freaking leave this video up. It'll probably get a strike, but that'll be good. Hopefully it gets a strike. If it doesn't, I'm gonna start asking questions. All right. Thank you. Take care, everyone. Have a good day. Thank you for joining, and great conversation.